Our first lecturer today is Himanshu Sharma. Himanshu is a dual MD PhD candidate in the medical scientist training program. He's working on understanding how various extracellular factors regulate the neural stem cell function in the developing brain. He was recently awarded a PHRMA Foundation predoctoral fellowship to translate some of his basic science advances to clinically relevant models of multiple sclerosis and some other white matter disorders. He expects to defend his dissertation and return to medical school in 2017. We were just talking about some surprisingly mutual interests. I should say I'm a political scientist, not at all in his field, but we both have an interest in neuroscience, and I'm very excited about hearing his talk. His talk today is Making Your Own Niche, Form and Function in the Developing Brain. From Pharmacology, Himanshu Sharma.
Thank you, Dr. Tabor. Thank you all for coming out this afternoon. So today I'd like to talk to you a little bit about, let me just put the presentation real quick. So today I'd like to talk to you a little bit about a project that I've been working on over in my lab in pharmacology, which relates to how form and function inform each other in the developing brain. And I think the story really begins about a decade ago when medicine itself seemed to be on the cusp of a new revolution. And at the center of all of the excitement was this entity known as a stem cell. And I think that this Time magazine cover here really sort of exemplifies the excitement that was in the air at the time, where it was titled, How the Coming Revolution in Stem Cells Could Save Your Life. And there was really this feeling, this palpable feeling that stem cells could usher in a new era of medicine where they could be used to treat all sorts of diseases and disorders ranging from diabetes to heart disease to Parkinson's. And over the years, as work sort of progressed in this field, it became sort of apparent that the path to turning stem cells into medicines was actually more complicated than many had expected. And so the development of therapeutics, of stem cell-based therapeutics in particular, has taken longer than we had really hoped initially. And I think this is also exemplified by the fact that Time magazine a couple of years later published a second cover article on stem cells, this time with a more ominous title, The Truth About Stem Cells, purporting to tell you what the hope and the hype was of these cells. So I think, the question really for a lot of people is, so what's the holdup? What happened? Why is it taking so long to develop a lot of these therapeutics? And I think in order to answer that question properly, we need to take a step back for a moment and understand exactly what a stem cell is. So a stem cell, very simply, is a cell that can do one of two things. It can either divide and replicate itself, making two stem cells, or it can go on and differentiate into many other cell types. So it can differentiate into a neuron, or a blood cell, or a muscle cell, for example. And essentially, that's it. And yet from this is where we all come from. This is a single fertilized egg alternating between these two actions gives rise to your body, my body, everybody on the planet comes from a single cell that just alternates between these two. And so I think it's really interesting because what the power that lies here is that if we can learn to harness and control how stem cells do this, we can treat diseases in a variety of ways. For example, we could take stem cells and repair cells that you've lost due to disease or injury. You could theoretically take a stem cell and grow an organ in a dish and transplant it into somebody who needs a version of that organ. And now with modern technology, one thing we can even imagine doing is taking some of these stem cells out of their body, fixing a genetic disease in a petri dish, and then putting it back into them, allowing them their cells to repopulate with the new corrected versions of their cells. And so for the longest time, the biggest challenge to developing stem cell-based therapies appeared to be the fact that stem cells, our best source for stem cells was embryonic tissue. And so that made the entire field of research both a politically and ethically charged and divisive field. But in 2006, a man by the name of Shinya Yamanaka made a huge breakthrough when he showed that you could take an adult cell, so a cell like a skin cell from near you, and by turning 4 molecular switches on in these cells, you could turn them into stem cells. And this sort of revolutionized the field because now with these, in this process, if we have these stem cells, we can go ahead and by giving them the appropriate signals, we can go ahead and differentiate them into cells, tissues, and various organs. And so now, today, the biggest challenge that we've come up with in translating stem cells into therapy has been the fact that this process, to turn a stem cell into a differentiated cell quickly, safely, and efficiently, is more difficult than we had anticipated. And so, just to reiterate, this is the biggest hurdle right now in delivering large-scale stem cell therapeutics. And so to tackle this problem, what we decided to do was to ask, how does nature do this? It happens every day in our bodies. Can we learn something from that process? And so in our bodies, stem cells aren't really found sort of sprinkled about at random. They actually exist in specialized microenvironments termed niches. And so these stem cell niches exist to help support the function and the development and the survival of these stem cells. And so one example you may be familiar with is that in your bone marrow is a stem cell niche that produces blood cells that go on to make your peripheral blood. And so in the sense, to use an analogy, we can think of the niche as the hometown for these stem cells. In the sense that some of the stem cells, they all grow up there, some stay there, some emigrate out and go on to do various things with their lives. So then we began to ask the question, so what's important in a home? And I just listed here a couple of things that are important to me personally. And on the left you can see the stuff in no particular order that's important to me to be happy and healthy. And then there's the sort of the bigger things in life. There's the direction. Who am I? What do I want to do when I grow up? And so we wondered if stem cells really need the same things from their home, from their niche. And it turns out they do. And the niche provides many of the same sort of aspects to a stem cell, of course, in slightly different ways. So, for example, stem cells have friends in their niche. They have other cells that support cells that interact with, support those cells. There's blood vessels that carry away the waste and bring in other signals and nutrients. They've got food in the form of oxygen, glucose, lipids, so on and so forth. They've got a place to rest, and this actually has an important effect on how they behave, in the sense that the topography, the stiffness, the elasticity of their physical location can play a big role in how they function and differentiate. They have, close related to that is sort of their shelter. And so what may not be as well appreciated is that these stem cells are also embedded in and interact with a network of extracellular proteins and molecules, collectively known as the extracellular matrix. Of course, there's the cops. They've got to keep the stem cells sort of in check, keep them from getting too crazy in their adolescent years. And finally, there's the mail. So essentially, there's messages that get sent to these both locally and from long distances around the body. And so what's really interesting about stem cells is that in addition to these sort of factors that I've discussed, they have a very specific spatial organization. And this orientation is also very important for the stem cells. So for example, you can see here that the orientation of the cells can help, in conjunction with some of these other factors, tell a cell whether to divide and make 2 stem cells, or alternately, when they divide, to go on and make 1 daughter cell and keep a stem cell. And so one of the niches that we're particularly interested in is found in the brain, and it's known as the subventricular zone, or the SVZ for short. And this is, similarly a microenvironment that houses neural stem cells that will go on to make up cells in the brain. And so if you were to take me right now and just sort of slice my head open like this, and you look at my brain, you would see something that looks kind of like this. And so deep inside the brain are these fluid-filled spaces, or ventricles, that are filled with a fluid known as CSF, cerebrospinal fluid. And this fluid, it's important for the regulation of the entire nervous system. It helps send messages through the nervous system. And if we zoom in on the wall of one of these ventricles, we would find our neural stem cell niche. And so the neural stem cell niche here, as you can see in this sort of artist's rendering, consists of the stem cells themselves, which are seen here in blue. And so these stem cells are sort of, they have processes that reach out deeper into the brain, and they have processes that reach and contact the cerebrospinal fluid. Additionally, one of the main niche support cells, or friends that they've got in the niche, are these ependymal cells, which we see here in yellow. And these cells have these projections known as cilia, and they extend into the CSF, and they beat in a coordinated fashion to propel the CSF and the signals it carries past these stem cells. And so the ependymal cells and the neural stem cells come together in this sort of pinwheel architecture, with the stem cells at the center, the processes of the stem cells at the center, and the ependymal cells sort of surrounding them. And then in this figure, we can also see some of the in green and in red here, some of the daughter cells that the stem cells produce. We can see the vasculature bringing blood flow to the niche, and that extracellular matrix, the network of extracellular molecules, proteins, and sugars that I mentioned earlier. And so if we were to sort of continue this analogy that I mentioned earlier, so if the entire niche as a whole is the village or the hometown, then we might think of each one of these individual pinwheel units as a single house. And as I mentioned, there's signals that are coming to all of these cells at all times. And so by extension of the analogy, we can think of some of these signals as mail that gets directed to the stem cells in these pinwheel structures. And so the question we decided to ask is, well, how do you send messages, or how do you send this mail to the house? Whether the messages are coming from the CSF, from the blood flow, or elsewhere. And in particular, we were very interested in one specific type of message. And we were interested in a molecule known as laminin. And the reason that we found this message particularly interesting was because laminin can serve as a message in and of itself. It's a member of the extracellular matrix, and it also can act as a sponge and present other signals to the neural stem cells. So we thought this was a very interesting candidate to understand how these stem cells and their niche support cells might respond to the signals that were being presented to them. So we decided to take a closer look at the subventricular zone architecture, and we were really excited to see that on the surface here, so if you imagine you're standing in this fluid-filled space and looking at the surface, you can see sort of the image you would see here. And so in green, we have the ependymal cells that are outlined here in yellow, and they surround the stem cells seen here in red. And I've sort of outlined the entire sort of pinwheel architecture to help you kind of to see that structure. And what we were really excited to see was that, in fact, we could find laminin, this messenger molecule, at the surface of this niche. And in fact, it had a very specific location. It was at the intersection between the neural stem cells and the ependymal cells. And so this raised the question to us whether, which came first, right? So did the message come there and say, build your sort of pinwheel house here? Or did the stem cells go ahead and build their pinwheels and then decide to put up some sort of mailbox so they could receive the messages later? And so we decided to, and so one of the really interesting things about the SVZ is that, so this is in an adult, at birth, none of this exists. It's just a flat sheet of stem cells. And over the first few weeks of life, the stem cells create this pinwheel architecture. They create the ependymal cells themselves. And in a very real sense, they're carving out their own niche that they're going to later exist. And so because this happens over the first few weeks of life, what we decided to do was to answer the question of which came first, to take snapshots over the developmental time period and see did the pinwheels form first, or did the messages come first. And what we found was actually really interesting to us, because we realized that some cells actually had laminin at birth, which isn't shown here, and these are sort of time points after birth. But even by the third day of postnatal life, you can see that, or hopefully you can see that the laminin is sort of present in this diffuse manner on the surface of some of these cells. And as the weeks progress, by the end of the first postnatal week, you can see that the laminin begins to sort of aggregate into these clusters. And by the end of the third week of postnatal life, so 21 days in, you can see that many of these much of this laminin is now found at the center of these pinwheels, which you can see sort of here. And this will continue until into adulthood when we reach a sort of final conformation. And so, it occurred to us that neural stem cells seem to be putting a lot of time and effort, devoting a lot of cellular machinery into orchestrating the movement of this laminin. First, it's diffuse on the surface of certain cells, and it begins to aggregate, and it takes weeks for this process to happen. And so, the reasonable question for us was, well, why? What's so important about these messages? And so what occurred to us also at the time was, well, if we think of laminin as the mail, if we think of this as the message that these cells are receiving, then one way to find out what's in the mailbox is to go around town and destroy all the mailboxes and see what happens. So that's actually exactly what we did. We found a receptor, we found a protein that's on the surface of these cells, but it spans the membrane. So it can interact with the laminin, which is on the outside of the cell, and transmit those messages into the cell, telling the cell how to respond to the presence or absence of the laminin. And so what we decided to do was we created a transgenic mouse in which all of the cells of the brain lost dystroglycan during embryonic development. And since, as I mentioned earlier, the ependymal cells and the adult stem cells arise from these neural stem cells, neither one of these cells could now read these laminin messages. And so first what we wanted to look at was, well, okay, if we destroy the mailbox, what happens to the mail? And so here again, we just see in this 3D reconstruction and also the same sort of surface view, in a normal mouse, at the end of the third week of postnatal life, you can see these laminin messages on the surface. And once we remove Dystroglycan, we find that, in fact, the messages are no longer there. So without the Dystroglycan receptor, the laminin messages are no longer present on the surface of this niche. So what? And so briefly, to just remind us, we've covered three things that stem cells can do, right? So a stem cell can divide and replicate itself. It can differentiate into various cell types. In this case, we're looking at brain cells. And during this time, it also creates a very specific and specialized three-dimensional architecture. So briefly, I just want to go through each one of these three things that we know stem cells do to see which of these aspects the laminin message was relevant for. And I'll just show one piece of data on each. So first, the ability of stem cells to proliferate, to divide and make more of themselves. So in the normal mouse, and so now we're looking at the same sort of cross-section if you were to look like this. In normal cells, and here you can see all the cells in blue, and on top of that you can see any cell that's actively dividing in yellow. So at birth, in the subventricular zone, you can see that a lot of these cells are dividing pretty rapidly, at a pretty rapid clip during this time. But over the first few days of life, this proliferation begins to die down. So they're shifting away from that sort of dividing and replicating phenotype. When we remove Dystroglycan, however, we found that at birth, they're dividing, although not quite as much as the cells in the normal mouse, but also that regulation, that down regulation of division appears to disappear, and so that they're dividing at the same rate at day 0 and at day 3 after birth. And because this is a science talk, I feel sort of compelled to at least present some quantification of this. And so you can see here in the normal mouse, in the white bars, from P0 to P3, or birth and 3 days after birth, you can see this drop in proliferation. But in the animals without Dystroglycan, that regulated change in proliferation is abolished. And of course, as I mentioned to you earlier, there's many different cells in the subventricular zone, and so we wanted to make sure that this was in fact in the stem cells, happening in the stem cell pool specifically, and we confirmed that the stem cells in fact did demonstrate this same phenotype. Okay, what about making daughter cells? So during the first postnatal week of life, this region of the brain generates a huge number of cells that will go on to myelinate the rest of the brain. And so the majority of the myelination of the forebrain, those cells are generated during this time period. And so we decided to look to see if this stem cell niche was really making those oligodendrocyte precursor cells at the same sort of clip. So here, again, in a normal mouse, we can see at day 3 that the SVZ, or the stem cell niche, is making some cells, but most of these cells, which is seen here in red, and these cells in red, however, are not really dividing at a rapid clip. When we remove Dystroglycan, we find that, and hopefully you can see this, that there's actually far more of these cells, which, these are rapidly migrating out of the SVZ once they're born. And furthermore, these cells will also continue to proliferate more, showing that the messages that these stem cells that Dystroglycan is sending isn't important just for stem cells, but also for some of these progeny cells to continue to function properly. And again, here's the quantification for the number of oligodendrocyte precursor cells made in the normal mouse versus the Dystroglycan deficient mouse, and as well as the percentage of those cells that are dividing in the normal mouse and the Dystroglycan deficient mouse. So the third thing, they make a very specific three-dimensional organization. And so now this time we're looking again at the surface of the neural stem cell niche in order to see that sort of pinwheel architecture to see if it really is disrupted. And so what you can see here in green are the cell borders, and in white, as if you recall, I mentioned that these ependymal cells have cilia, right? So they have these projections that push the CSF past them. And we immediately noticed just from looking at it that not only was the pinwheel architecture, the architecture of the house sort of disrupted, but the cilia themselves appeared to look sort of strange. And so we decided to try and get a better handle on this by understanding what was going on with the disruptions in the cilia. And so briefly we measured, I'll show you measurements of two things that we've looked at, which is how far away the cilia are from the center of the cell, and in the normal wild type, they're usually sort of polarized in one particular direction, and also the size of these patches of cilia. And so we find that that the displacement is higher, by which I mean these cilia appear to be sort of scrunched up along the border of the cells, right? So they're not really in their normal location. And additionally, the size of these patches is smaller, which implies perhaps that these cells are actually making fewer cilia. And as I mentioned, these cilia are important for regulating various things in the brain, including sending signals across the stem cells. And as a result, we found certain sort of defects in development that can be attributed to ciliary defects such as hydrocephalus, for example. Okay, so then we decided to ask, so we've removed Dystroglycan, we've removed the mailbox from all of the cells in town, from all the houses in town. And so everything's screwed up. We see changes in both the ependymal cells and the stem cells and the cells that are being produced. So, but as we can see from where the laminin was, where the messages were being delivered, that both the ependymal cells and the stem cells could theoretically be reading it. So which one was it? Which cell type are these messages important for? And in order to answer this question, we created two new transgenic lines. And in the first, we were able to remove the ability of the stem cells to get to read the laminin messages, and we did this shortly after birth, so that the ependymal cells, which arise from the stem cells, still had their dystroglycan. They could still receive and respond to any laminin message, but now only the stem cells after birth would no longer be able to get these messages. And so you can see here, again, in cross-section, so you can see these blue, the stem cells are in blue. Here we've marked in green. So these are sort of the normal stem cells in the mouse at the end of the first week of life. And when we remove dystroglycan from these cells specifically, we find that we actually have a lot more stem cells. And so this is a stem cell sort of specific phenotype in the brain. So what about ependymal cells? We generated, as I mentioned, we generated a second transgenic line where we removed it only from the ependymal cells. So now the stem cells can receive the messages and respond to them appropriately so long as they're there, but the ependymal cells can't. Does this change the ependymal cell behavior? So this time, again, we looked at the sort of surface of it to look more carefully at the ependymal cells. And again, the stem cells here are marked in green, and you can see in white the laminin message. So the laminin is still there in this case. So this is in the normal mouse. So when we remove dystroglycan from only the ependymal cells, allowing the stem cells to still respond to the messages, we can see that there's actually far fewer stem cells now present in the niche. And in fact, while there's less laminin, indicating that the ependymal cell dystroglycan is still important for some of it, the messages are still there. So it's not as though the stem cells can't respond to these messages. It's in fact the ependymal cells being unable to respond to the stem cells is somehow changing the effect of the stem cells' ability to sort of stay in their niche in a happy and healthy sort of way. So what does this mean? What have we sort of learned so far from these interrogations? So one, I think the take-home message here is that the local environment is critical to how stem cells respond to messages. So it's not just the message. In fact, are they competent to read the message? Do they have the proper receptors at the proper time to read the messages we're sending them? Who else is reading the message? So what are the other niche support cells that are reading the message, and how does that affect them? And I think these two lead to the third, which what is the context, right? So in the last image I showed you, ependymal cells no longer are getting the message, and somehow they're talking through a non-laminin mediated way, it appears, to the stem cells. And so I think all of these lessons sort of give us a point of the way forward to helping improve and get better at this step. And I think there's two sort of major directions we can take these lessons in. First, can we make this process more effective? So if we're taking a stem cell out of the body and we're trying to make a differentiated cell, can we take a step back and consider, well, what are all the other messages they're receiving? Do they need certain stem cells, certain support cells? Do they need a particular architecture? Do they need a particular orientation in order to differentiate properly and effectively? And additionally, I think we've also seen that the organization of the stem cell niche is very intricate, and these cells have a very complex way of interacting with each other. So can we take advantage of the stem cell niches that are already in our body using the sort of context-dependent signaling to understand how we can harness those niches we have and make them more effective at responding to injury and repairing various insults to the body. And so with those thoughts, I'd like to just say thank you. I want to thank two people in particular, our lab's fearless leader, Dr. Calignato, and also one of the previous graduate students in my lab, Dr. McClenahan, now at Cambridge, who did a lot of the, who generated the initial mice and did a lot of the initial characterization of these mice. And with that, I'd be happy to take any questions.